Oh, yeah. Good morning. The talk will not be very long because it's all on the newer drugs. So we know about two things. Once you place a stent, what happens? So one, there is a dissections, exposure of the media, which causes a thrombosis. And second, the stunt strets which are there. So they actually stimulates the vascular smooth muscles to cause you neomental hypoplasia. So what we are talking about is to prevent these two very important things in our practice. So we saw this slide yesterday, the PTX and sirolimus. So I'm not going to go in detail here. And we now know why we need neoreagents, because there is a limitation of the drugs what we have. So the important drug paclitaxel has got some side effects, and sirolimus is not a very, very potent drug, and there is not a good drug distribution because of the lipid profile it has. So we need an improved efficacy and safety profile of the drugs. And of course, there is a lesion problems like calcifications and smaller vessels. We need the neoreagents who has better tissue retention, minimize systemic toxicity, enhanced anti-inflammatory and reparative properties, and compatibility with advanced platforms. There are a huge amount of advanced platform which has come up. So look into these classifications. So we have heard just now, so we got sirolimus, everolimus, jotrolimus, and we also got biolimus A9, which is also being used now extensively. So we have got calcineurin inhibitors like tacrolimus and the punicrolimus, which I have never heard of before. So I read it for Shekhar. So microtubule stabilizer, we got paclitaxel, which we use routinely. And there is something new which has come up is, I don't know anybody who have heard is EPC capture. Anybody has heard of EPC capture? This is endothelial progenitor cell captures. So what is a very interesting technology. We'll go through this, okay? So what is the mechanism benefits of neoreagents? Is anti-proliferative effects. We don't want the vascular smooth muscles to come there. We want the drug to go in, and we also want a targeted action. But at the same time, we also want the new intima to come on the stent. Because if the new intima is not going to come, you're going to have an earlier thrombosis. So these are the basically 5 technologies where the world is going. One is the biolimus, where we have widely adopted in some regions now in the world. Jotrolimus is being incorporated in the Resolute Onyx stents now. And novolimus is in the early phase clinical trials. And the gene-based therapies and combination therapies, they are in the experimental stages at present. So let's see the 7 to 8 things which I have talked about. So let's talk about biolimus, what it is. We haven't heard about this in 1.5 days. So it's important to learn about newer technologies. So it's a biodegradable polymer. It's a semi-synthetic analog of sirolimus. You see, most of the newer drugs which are coming up, they are not based on paclitaxel. So they are all based on sirolimus. It has got higher lipophilicity, which is important because the faster tissue uptake and shorter systemic exposures. And there have been 3-4 studies which I have mentioned here. I don't want to go in detail about that. And all showed that in some study it showed is equivalent to sirolimus or it is better than sirolimus. And it reduces inflammation and faster healing time. So why are we not using it? Because it has got a longer term data we still don't have. Another drug is coming up is a jotrolimus. So it is also a rapamycin derivative, similar to sirolimus, and it also has got potent anti-proliferative effects. It has been actually integrated into the Resolute Onyx stent now, and it has got a shorter drug elution period, which is very important for us, and minimal delayed healing, but still under analysis, and the data is being collected with this drug. Then another drug which has been studied is a novolimus. It's another drug which has got enhanced lipophilicity. So the drug can go inside very fast inside the vessels. So early clinical trials are being done, tested in biodegradable polymers. So basically, we don't want the metal to be there. So most of the newer technologies are going into biodegradable polymers with a faster drugs going inside and not very strong anti-proliferative effects, but and less cytotoxic. So still limited clinical adoption because of lack of large clinical trials. Another one is an everolimus. Now this is being used now, and as a routine practice, it's being used in India also. And I think future studies are warranted to compare the results with the sirolimus drugs. Then a tacrolimus based, which is a calcineurin inhibition drug. So this is not based on sirolimus, though it is limus, but it works in a different ways, and it actually binds to FKBP12 protein. So this chemical pathway you can understand yourself or read it on the internet, and actually it blocks the smooth muscle cells. So coming to the point, and it's working on that. Similarly, we got pimocrolimus, same way it works like that. Now this drug actually was used by the dermatologist for eczema, and it has been picked up from there and being used as a vascular smooth muscle inhibitor in the preclinical stage. So now there are another drugs which are coming up called as euminrolimus and the tiolimus. They are all very potent drugs with the limited side effects, and still being studied in the literature, and a lot of trials are being done with these drugs. So a lot of money has actually been put in into these technologies. I don't think the amount of money you see from the internet which has been put in all these technologies is not going to go away, because I think people have got spent a lot of money into this. They work or not, we will see. Now this is another very important drug, a DO drug. Means 1:9 ratio of paclitaxel and sirolimus. And it's been, this actually has been used now. It encapsulated in the biodegradable nanoparticles, prolonged drug distributions, lesser cytotoxic effects of the paclitaxel, and good points about the sirolimus combined together. So this balloon is being experimented and being studied. So combination of both the drugs. Then come the gene-based therapies which are being included, that you actually have the microRNAs attached to the technology, and it actually gets into the vessel wall and modify the vascular healing. So of course there are limitations because of regulatory complexities and delivery challenges, and still into the preclinical stage. Now there are a lot of combination therapies which are being used now. So I'm going to go about that. Now one stent is being developed in India actually. It's been heparin and sirolimus combination. So it's been in the coronaries, not in the peripheries. It's been experimented and the trials are going on. Then there is another compound called the genestin and sirolimus combinations is being experimented on the stents and being studied. Now this is a very interesting technology, EPC, which I was talking about. So what do is you take a stainless steel or cobalt chromium stent, you put in a CD4 antibodies on the top of that, and you place the stent inside the vessel. So CD4 antibodies actually will capture the endothelial progenitor cells from the circulation, and actually all these cells will encourage rapid endothelialization, what we want, so that you don't get an earlier stent thrombosis, and they will not actually initiate a vascular smooth muscle proliferation. So this is also being studied in the world, and a lot of studies are being going on. Then there is another stent with an anti-VEGF drug from UK, which there actually it decreases the formation of neovasovascerums and actually cause plaque stability. So it works on a different mechanism. So this is also another way to look at into it, so that you don't have a huge problem with the neointimal hyperplasia. So we know about the PLLA stents, which we talked about, is being coated with various drugs now and being studied. And there is another one which is very important, is Riva stent. It's a tyrosine-derived polycarbonate. It's radio opaque. You know, what happens is the biodegradable stents, most of these stents, you cannot see them, and they are radiolucent. So you just have to have depended on the markers at the ends to deploy it. So sometimes their deployment becomes a difficulty. Then there is a magnesium-based stents which are being studied now, Bear stents were not very different from the normal bear stents, but now they are being combined with the drugs like the sirolimus and everolimus. So much is there. Every company and every country, I think, is trying to prove that this toy is better than the other. And I think it was going to happen the same way. There no comparison of the toys will happen, and everybody will be confused at the end which one to use, and everybody will be using it according to the availability and the cost. So what are the limitations? So biological challenges is a very important, because the drug elution kinetics or calcified lesions, smaller vessels, how are we going to do it? Then there is the clinical evidence. There is not lack of large scale, there is a lack of large scale randomized control trial. We saw in 1.5 days, 150, 100 patients, 80 patients, and that's why we are all confused at the end of 1.5 days. Should we use it or should we don't use it? Unanticipated side effects, which we don't know. Extensive testing and approvals is a huge amount of money you have to put in. Regulatory hurdles are there. And cost of the balloons in our country especially. Technological barriers, which I have talked about, is a very big problem, especially like a sirolimus. And there is a market resistance, because the drugs, the companies who have already invested so much money on sirolimus and paclitaxel, and you saw Australia still using paclitaxel coated balloons everywhere. So I think it's so complicated, and it's not only the studies. I think it's a lot of money driven, market driven, company driven, a lot of physician driven, a lot of other factors also comes into the picture before we understand everything. So the newer agent, in summary, this is the last slide sort of, in developments are like a biolimus, which is there already, is being used in many countries. Jatrolimus is also being started using in many countries now, and it is a rapid metabolism antiproliferative. Novolimus and sirolimus derivatives and gene-based therapies and the combinations, they are all into the experimental and the preclinical stage, and maybe in the next 1, 2, 3 years, we will hear about them as we go along. So in conclusion, I will say newer agents holds promises for improved efficacy and safety, and overcoming all these parameters is very critical, and multidisciplinary collaborations and advanced trials, and I think physician-led trials are more important, especially what Shekhar had put in yesterday, Basel III trial. I think this is a very important trial, because what you see, all company-driven trials shows benefits, all physician-driven trials shows no benefits. So it's a very, very, very important statement, and I think we need more trials which are physician-run, not company-driven, and to know does they work or not. Thank you.